shortly introduce Dr. Andreano. But first of all, I would like to introduce myself. My name is Lars Frelin. I'm the chair of the Outreach and Public Engagement Committee within the International Society for Vaccines, who are the organizers of the webinar series. And first of all, I would like to have a few words of advertisement of the International Society for Vaccines, which is a global not-for-profit organization that aims to encourage, establish, and promote the development and use of vaccines to prevent and control infections and non-infectious diseases in animals and humans. And if you are interested to become a member, you can just go to our website and register for a membership. And we have a number of different benefits of being a member, so I can highly encourage all of you that are not a member today to become one tomorrow. I also would like to highlight our annual Congress that will take place in October 21st to 23rd in Seoul, South Korea. And the announcement is already out, so you can already now submit abstracts and also register for the annual Congress that will be taking place at the Dragon City Hotel in Seoul. So I would like to welcome all of you to this annual Congress. Also, we have a next webinar coming up in May, the 22nd of May. It will be a talk by Dr. Montemoli from University of Siena, who will talk about serology for influenza and other respiratory disease vaccines. So I can encourage you to make a note about the date of the webinar, and you can also find all information on the ISV webpage. Another important webinar that will take place is a webinar organized by the mentorship program within the International Society for Vaccines, and the host will be Dr. Schuchat and Boyer, who will organize a mentorship program that is open for both if you would like to become a mentee or if you're looking for a mentor. You can join this information seminar on May 2nd. Also, information about this is available on the ISV website. And I also would like to highlight that the International Society for Vaccines are very, very happy for the support from our different sponsors. And I would like to thank all of the sponsors for the contributions that will make it possible for us to perform our mission and our different programs. And if you also want, you can follow us on our social media platforms. We are available on Facebook, LinkedIn, YouTube, and also Instagram. And if you scan this QR code, you will come directly to our LinkedIn account. So please follow us for more updates and information. And then I'm very happy to introduce to you the speaker of today, Dr. Andreano from Toscana Life Sciences in Italy, who will talk about the evolution of the B-cell response in COVID-19 mRNA vaccination hybrid immunity. And Dr. Andreano did his bachelor in biotechnology at the University of Siena. He also did his master in immunobiology at Newcastle University, and then he moved back to Italy and performed his PhD at the University of Siena, where he now performs vaccine research in areas like COVID-19, for example. So we are very happy to have you here today, Dr. Andreano, and looking forward to your presentation. A few housekeeping rules is that you can pose questions in the Q&A section in the webinar, and you can also raise your hand by the end, and I will unmute you so that you can ask questions directly to Dr. Andreano. So most welcome, and looking forward to your speech.
Thank you very much. Thank you very much, Lars. I'm just going to share my slides. Right. Okay. You should see the presentation, right?
Yes, it's perfect.
Okay. So again, thank you very much for the kind introduction. And before starting, I really would like to take this opportunity to thank Professor Ted Ross and Dr. Giuseppe Sauto for their kind invitation and for giving me the possibility to share some of the work that we are doing at the Monoclonal Antibody Discovery Lab, or MAD Lab, based at Fondazione Toscana Life Sciences, Siena, Italy. And today, as you can read from the title of this presentation, I will talk about the evolution of the B-cell response in COVID-19, mRNA vaccination, and hybrid immunity. So I don't think I need to introduce this concept much, but we are all aware about the dramatic economic and human life loss that the COVID-19 pandemic has led. There has been at least 700 million cases of infection worldwide, but the estimated will be much more, and over 7 million deaths in the last 4 years. The peculiarity of this virus is that it keeps evolving over time, over and over again. Indeed, since 2020, we have over 70 major variants that have been identified. And this virus keeps evolving to evade natural and vaccine-induced immunity, as well as human monoclonal antibodies that have been developed for both therapeutics and prophylactic interventions. So it is important, it is imperative to develop enduring vaccines and therapeutics. And to do so, it is important to study the immune system and how it is possible that the immune system can tackle this virus and all the variants that emerge from it. So just gonna take this down. So our laboratory focuses, as I said, mainly on human monoclonal antibodies and on B cells, especially. So that's the major focus of our lab. And what we study, as you know, the SARS-CoV-2 spike protein is the main target of neutralizing antibodies. And every year since 2020, basically, the virus keeps adding mutation over mutation on the spike protein. And the higher the levels of mutation, and the higher the levels of immune evasion and antibody evasion that this virus leads. So we study the response, the antibody response of different donors, of different cohorts, and how the B-cell compartment evolved to control the different variants that have emerged so far. So during the last 3 years, pretty much, we investigated longitudinally 4 different cohorts. The first cohort here are people that got vaccinated just with 2 mRNA vaccine doses, and we call SN2, so 2 seronegative, so no previous infection and just 2 mRNA vaccine doses. And we had 5 people like this. And then subsequently, 4 of these donors were re-enrolled after a year when they got a 3rd booster dose. While here in the hybrid immunity cohort, we have different immunological background. We had initially people that got a Wuhan infection, and then later on, these people got 2 mRNA vaccine doses as boosters. Some of these people, 2 of these people were re-enrolled later on when they got a 3rd booster mRNA vaccine dose, and then an Omicron breakthrough infections, possibly by BA.2 and BA.5, based on the year and period from which these people were infected. So we studied at single cell level the B-cell response of all of these cohorts. And now we did that, basically we enroll the people, we collect their blood, we usually take around 100, 150 ml of blood, from which we can isolate the PBNCs. PBNCs are then used to perform single cell sorting of antigen-specific memory B cells, and in our case, we use the spike protein as antigen bait for their sorting. Once single cell stores it, these cells are incubated for a couple of weeks, so they can naturally produce and release antibodies within the supernatant. And once this occurred, then we take all the supernatant and we screen in BSL-3 with a live virus, all the antibodies naturally produced to identify the neutralizing ones. So once we have our neutralizing antibodies, we performed analysis that cover both the Fab region and the FC portions. Concerning the Fab region, we evaluate different aspects of our monoclonal antibodies, specifically the breadth of neutralization, as we use all the different variants that emerged during the last years. We also mapped the epitope region targeted by our neutralizing antibodies. And finally, we perform B-cell repertoire analysis to understand the genetic signature and profiles of the antibodies that are evolved after vaccination and infection. The other portion is the FC that we also take into account. So we don't just focus on the neutralization, but we also evaluate the phagocytosis and complement deposition activity mediated by these neutralizing antibodies. So how the antibodies play also with the other cell compartments of the immune system. And in the last 3 years, pretty much, basically, we built the panel of almost 1,000 neutralizing monoclonal antibodies that as soon as the new variants emerge, we are ready to test right away against the new variants. So the first question that we asked to ourselves was whether mature B cells and antibodies were able to protect from the different Omicron variants that were emerging in the last years. So we performed a neutralizing assay. I'm just going to try to remove this. Otherwise, this is going to cover up. I don't know how to do this. Okay, no. So first of all, we tried to understand how the neutralization worked in the first cohort, which were these 2 mRNA vaccine doses. From these people, we managed to isolate a total of 52 neutralizing maps, which were all tested by neutralization against all the different variants that you see here in the graph below. So as you can see from this graph, basically, except for one antibody that was able to neutralize with some medium potency, BF.7 and BQ.1.1, none of the antibodies were able to neutralize the variants, including the latest one. Then we analyze also people from the 3 mRNA vaccine doses. And basically, from these people, we isolated 206 neutralizing antibodies. And we observed different scenarios here. So as you can see from the graph, after a third booster dose, we see the increase of antibodies able to cover and neutralize also the newest variants, including BA.2.86, with the neutralization coverage ranging from 3.4 to 17%. Another important note is that the antibodies found neutralizing the 3 mRNA vaccine doses weren't that potent. So they were neutralizing, but their potency was ranging between 1 microgram and over 10 microgram per mil. So next, we performed the same analysis also for people with hybrid immunity. From people with hybrid immunity, we isolated 224 neutralizing mAbs. Here we observe this scenario similar to the 3 mRNA vaccine doses, where we see a coverage ranging from between 3.6 and 13%. But interestingly, in the hybrid immunity cohort, we didn't observe any neutralizing antibodies able to cover the latest 3 variants, XBB 1.5, AG 5.11, and BA.2.86. In the super hybrid immunity cohorts, where we managed to isolate 417 neutralizing mAbs, we observed a huge boost in the neutralizing response against the latest variant. Here, neutralization coverage was ranging between 6 and 62% against the latest variants. Interestingly, this was the only cohort that showed not only broad neutralization, but also to produce antibodies with a strong neutralization potency, and some of which neutralize viruses also with a neutralization potency below 10 nanograms per mil. So in addition to the neutralization, as I said, we also study the FC function of these antibodies. And here, if you remember, with the two mRNA vaccine doses, we really didn't have any neutralization against the variants XBB.1.5 and BA.2.86. So no neutralization there. Despite that, we observed that some very low percentage, very low frequency of antibodies retained some phagocytosis and complement deposition activity. So just 1 to 7%. When we evaluated the same thing with third mRNA vaccine doses, then we observed again a boost, not only neutralization, but also in the Fc function. Indeed, you can see here that we had roughly 2 to 7% of neutralization against BA.2.86 and XBB respectively. But when we observed the Fc function of this antibody, we observed an increase spanning from 15 to over 35%. We did the same analysis also for hybrid immunity and people with super hybrid immunity. As for hybrid immunity, again, no neutralization was observed against these two latest variants, but a little bit of Fc functions were still retained from neutralizing antibodies recovered from this cohort. And again, when we analyzed the super hybrid immunity cohort, we observed once again this incredible boost in the response, not also in terms of neutralization, but also in terms of Fc function, which was now ranging between 17 and over 30%. So not only neutralization, but the booster dose, either in the 3 mRNA vaccine doses or in the super hybrid immunity, expanded also, increases also the ability of antibodies to induce Fc functions. So now we know about the neutralization Fc activities of all these panel of 1,000 antibodies isolated from the 4 cohorts. Next thing that we wanted to know was which were the portion of the spike or the spike protein that were able to mediate cross protection. So to do that, we classified our monoclonal antibodies into 5 different classes. So based on their ability to target the RBD, and whether they were targeting the class 1-2 epitope region here on the top of the RBD, class 3 in the right flank of the RBD, and class 4 on the left flank of the RBD. Then we had antibody that did not compete with, still targeting the RBD, but not competing with the 3 classes here. And then we had antibodies targeting the NTD region. So here we started trying to understand the regions targeted by antibodies that mediate cross protection in the 2 and 3 mRNA vaccine doses. Well, if you remember, we didn't have neutralizing antibodies in the 2 mRNA vaccine doses and against the different variants. So here we really couldn't make any sort of analysis. The only thing that we can highlight is that in antibodies targeting, neutralizing the Wuhan virus, so the original virus, the majority of the antibodies isolated from this cohort were targeted in class 1-2 epitope regions on the top part of the RBD. Differently, when we analyze the 3 mRNA vaccine doses here, then we observe different trends. So initially, if you observe here the pie chart, distribution of these antibodies, the majority of antibodies found to neutralize after a 3rd booster dose still were class 1-2 for Wuhan. And these antibodies were the most cross-reactive also against the different variants. Indeed, they accounted for almost 50% in average of the antibodies covering all the different variants that have been tested here. When we did the same for hybrid and super hybrid immunity, we observed different profiles. Indeed, if you see here the hybrid immunity cohorts, we see that at the beginning against Wuhan, the class 1-2 antibodies were still the most abundant in the hybrid immunity cohort. But cross protection against the different variants were mediated by antibodies targeting different portions of the RBD. Specifically, we had the majority of the antibodies with either the class 3, so here on the right flanking side of the RBD, or the NTD. Then when we analyzed the super hybrid immunity cohort, we observed a switch in the targeted region of these antibodies. Indeed, while hybrid immunity was mainly targeting class 3 and NTD, super hybrid immunity came back to the class 1-2. So they were maturing again class 1-2 antibodies. They were restoring class 1-2 antibodies to covering also the newest variants. And this profile was very much in line with what we have observed in the third mRNA vaccination. So now we know that super hybrid immunity gives the strongest neutralization on Fc functions. We know that maturation of the antibody response pushes towards class 1 epitope region. And so the next question that we had was, which were the predominant B-cell germlines that were encoding for these cross-protective antibodies. So here we started by analyzing the antibody response in the hybrid immunity and super hybrid immunity. Here it is shown in what is called a network plot, where basically all the different antibodies in the two different cohorts were pulled together and then clustered based on their sequence similarity. The centroid, so the gray dot that you see, is basically the germline line, and then based on the distance indicates how far mature is the evolution of the antibody compared to the germlines. And links between centroids means that there is a shared properties found in the different centroids. And as the antibodies colored in pink will be the one from the hybrid immunity, while the dark red are antibodies from the super hybrid immunity. So as you can see, we found either antibody that were basically expanded, clusters that were expanded from the hybrid to super hybrid immunity, as for the light purple color that you see here, or up here, or down here. And we also see some clone types, basically some clusters that were expanded that found only in the super hybrid immunity cohorts, so that we did not observe in the hybrid immunity alone. So they needed a booster to be expanded. So then we took these clusters, either expanded or unique clusters from the super hybrid immunity, and we wanted to understand their ability to cross-neutralize all these newest variants. So as you can see from these heat maps here, we observed that there were some specific germlines encoding like the 353, 36, encoded by the 353 and 366 V gene with different J genes. They were the only one basically giving from the expanded clones that were able to cross-protect the latest variants, including BA.2.86. In addition, we also find one germline, which is this 510-1, paired with the J gene 41, that was able to cover also the BA.2.86 variants. Interestingly, is that these 353, 366 found to be cross-reactive against the latest variants were also the most common and the most publicly shared at the beginning of the COVID-19 pandemic. Indeed, these germlines were found in people that got COVID-19 infection, SARS-CoV-2 infection, or RNA vaccination, or with hybrid immunity. They were always there. And these were the germlines that were restored by the original antigenic scene led by the Wuhan virus. Then we decided to compare the repertoire that from the two most evolved and matured B-cell response that we have in our hands, which are people with 3 mRNA vaccine doses and people with super hybrid immunity. So we decided to see if there was any convergence between these two. And we basically observed that there is a huge convergence of the B-cell response, the B-cell repertoire shared by people with 3 mRNA vaccine doses and super hybrid immunity. And again, we found that the shared one were the 366, 353, and then also the 169 paired with different J genes and the 158, 31. So again, independently from the priming that these cohorts have had, vaccination for the 3 mRNA vaccine doses and infection with for the super hybrid immunity, they were still expanding the same B cell germlines. We then decided to compare the neutralization activity of the expanded germlines and shared germlines between 3 mRNA vaccine doses and super hybrid immunity. And as you can see from these heat maps, there are different profiles of neutralization found. Indeed, for instance, the most cross-neutralizing ones that we identified for the 3 mRNA vaccine doses were these 16941 germlines, which was very low potent and low cross-neutralizing in the super hybrid immunity cohorts. Differently, we observed that the 353, 356, 366 germlines found in the super hybrid immunity cohorts were restored and were also the most cross-protective against all the newest Omicron variants. So we now know that the antigenic scene pushed by the Wuhan, both infection and vaccination is there, and with additional mRNA vaccine doses and Omicron breakthrough infection in the super hybrid immunity cohorts, the immune system will still restore these 353 and 366 germlines to make them able to cross-protect from the newest Omicron variants. So because of this latest restoring these B cell germlines, our next question was how it was possible that the JN.1 variant was able to evade so brutally our immune response and quickly spread and conquer the world. So as you know, JN.1, which was detected latest 2023 and basically spread globally since December 2020, spiked since December 2023, and now it still is with these sublineages the most predominant variants worldwide. And interestingly, while the antibodies, restore antibodies from the super hybrid immunity cohort were able to protect from the Omicron BA.286, the ability of the virus to spread globally showed us that this restored B cell response was not able to cover these newest variants. Despite JN.1 differs only for one substitution in the RBD, which is the L455S. So again, we took all our panel of 1,000 neutralizing antibodies, almost 1,000 neutralizing antibodies, and we just run neutralization assay again to understand how these antibodies were now able to cross-react against the JN.1 variant. And as you can see from the graph here, basically, none of the antibodies in 2 mRNA vaccine doses, 3 mRNA vaccine doses, and hybrid immunity were able to cover JN.1. Differently, people with super hybrid immunity were the only cohort that were showing, even if a very low percentage, just 5.5%, were able to cover a little bit these new variants. And again, we decided, let's check also for the Fc functions of these monoclonal antibodies. We didn't test all of them, but we just test those that retain binding to the spike protein of JN.1. And we observed that despite no neutralization was observed for the 2, 3 mRNA vaccine doses and hybrid immunity, levels of phagocytosis and complement deposition were still detectable. And this is true for all the cohorts, including the ones that were where we didn't find neutralizing antibodies. So the 2, 3 mRNA vaccine doses and hybrid immunity. Then we wanted to understand again which were the classes of antibodies that were mediating cross-protection in the super hybrid immunity cohorts against JN.1. And as you can see here, basically again, these pie charts, the majority of antibodies that were able to cross-protect against BA.2.86 were the class 1-2. Over 60% of the antibodies from class 1-2 were responsible for cross-neutralization against BA.2.86. While over 20% were those targeting the class 3, so the right flanking side of the RBD. If now we observe the antibodies profile for JN.1, again, we see a completely change. Indeed, almost half of the antibodies were now covering for JN.1, so just above 30% of the antibodies were now able, of class 1-2 antibodies were now able to neutralize JN.1, while antibodies from class 3 almost doubled, and almost 50% of antibodies covering this variant were now able to neutralize, were now targeting the class 3 epitope region. So again, now we know the neutralization for JN.1 only found in superhybrid immunity. We know that class 1-2 lost the majority of their activity against JN.1, while class 3 were now the main neutralizing. And we wanted to understand also the ability of these antibodies to induce phagocytosis and complement deposition. So we evaluated the frequency of these antibodies to evaluate all the three functionalities. So again, if we observe neutralization, we had half of the antibodies in class 1-2 that lost basically their neutralization activity compared to the ancestral BA.286. And the same thing was noticed also for phagocytosis and complement deposition. So these antibodies, class 1-2 antibodies, not only lost their ability to neutralize, but also their ability to induce any Fc function. Conversely, antibodies targeting the class 3 epitope region not only doubled in terms of frequency for neutralization, but also in terms of phagocytosis and complement deposition. And this is true also for class 4 antibodies, but these are so rare, so they're a lower portion. But as you can see, also class 4 doubled not only in terms of neutralization, but also in phagocytosis, while complement deposition was never observed. So we went and checked also the B-cell germlines that were encoding for these different classes of antibodies. And we found out that basically the 353 and 366 that were restored against BA.2.86 in the superhybrid immunity cohort, basically over 90% of these antibodies were now evaded against JN.1. While the class 3, if you remember the germlines 510-1, which was the unique germline found in the superhybrid immunity cohort and able to neutralize also BA.2.86, was now the most abundant and predominant in the neutralization response against JN.1. Finally, we observed this trend basically, where at the beginning of the pandemic, we had these 353, 366 germlines that were the most potent, then they were restored, then they will start to be very active and functional against the newest variants. But then with the advancement of newest variants like AG.1.1 that start losing potency, they were able to cover the variants BA.2.86, therefore blocking the expansion and the infection of these variants, while with the introduction of just 1 point mutation, the over 90% of these germlines were evaded against JN.1. While a different profile, a different scenario was observed for the class 3 antibodies encoded for the 510-1 germlines, where basically they were expanded with the BA.2, BA.5 Omicron infection, but they were quickly evaded, and now they got a little bit restored, and now they are the most abundant in the neutralization for JN.1. So the last thing that we asked to ourselves was how it was possible that JN.1 was able to rapidly evade the response of the restored germlines 353 and 366. So going back to the literature in 2020, 2021, we found out that there were two major motifs that were described to be pivotal for the binding and neutralization activity of antibodies encoded for 353. One motif was in the CDR2, and one motif was in the CDR1 of the heavy chain, and it is these three NY motif as residue 32 and 33. And this residue, especially the N residue in this motif, was shown to basically stabilize the Y33, and Y30 and Y33 was able then to bind L455 on the RPD. Not only this, but Y33 needed this binding to this set of residues here and the L455 to form an hydrophobic cage that allowed the binding of Y33. So we interrogated basically our antibodies encoded from the 353, 366 germlines from the superhybrid immunity, and we had 37 antibodies from these two germlines. And as you can see from this logo plot, all of them, almost all of them actually, had this NY motif in them, meaning that this motif still is, despite Omicron breakthrough infection, very much conserved in all antibodies encoded by these germlines. So when we perform molecular dynamics with all of our antibodies with JN.1 and Wuhan RBD, we observe that all of our CDR with tyrosine 33, Y33 in the CDR1 were fitting perfectly in this hydrophobic cage of Wuhan RBD. With the L2S substitution in position 455 of the RBD, basically disrupts this hydrophobic cage, and the tyrosine 33 was now not able to anchor anymore in this hydrophobic cage, losing their ability to bind, neutralize, and induce Fc functions. So to conclude this presentation, there are three key points, three key messages that I would like to highlight. So superhybrid immunity, the power of superhybrid immunity. So this has been shown, and we observe the same thing when you got hybrid immunization, not only because of vaccine and infection, but also heterogeneous with different variants, in this case, Omicron variants, really boosts neutralization and Fc functions. The second key point is the importance of the antigenic scene in driving this convergent B-cell response. Indeed, independently from homogeneous booster when you got 2 or 3 mRNA vaccine doses, but always with the same spike protein, or if you get heterogeneous immunity when you get different exposure to another S protein, you will always have this scene there of the 353, 366 germlines, which are ready to be restored to neutralize the new virus. And the last message is this. So in the last 3 to 4 years, the COVID-19 pandemic really allowed us to track in real life the evolution not only of SARS-CoV-2, but also of the antibody response. And it is clear that there is this unceasing interplay between these two systems that continues to evolve over and over again. And it will be interesting to see over time if this will stop and virus and antibodies will find the balance, or there's going to be a new evolution of both the virus or the antibody response, or they will continue to restore and chase one another. And with this, I would like to thank my mentor, Professor Rino Rapoli, and of course, all the team that have performed this incredible work, and especially Ida Pacello and Giuseppe Maccari, which have been the two lead authors of the work that I've just shown you. And of course, the whole Mad Lab and the hospital in Siena, without which we couldn't have all the donors and that we have analyzed in our studies. And with this, I'll close my presentation, and I'm happy to take any questions.
Thank you very much, Dr. Andreano, for a very nice presentation and a very impressive work that you have performed together with your team. So congratulations. So we have a few questions in the chat, and I also know that I think someone also raised their hand, but maybe we could start with one question in the Q&A, the chat function. And it's a question from Marcella. How are neutralizing antibodies measured? If you could explain that shortly.
Sure. We perform, it is a cytopathic effect-based neutralization assay. So basically, we take the antibodies naturally produced, we co-incubate the antibodies with virus, live virus. All the experiments are performed with live virus in BSL-3 laboratories. And then basically we moved that on 0E6 cell lines, which are very well used for this sort of neutralization assay. And after 3 to 4 days, based on the variants, it is quite variable, we observed the cytopathic effect of the virus. If there is no cytopathic effect, the antibody is protective. If there is cytopathic effect, the antibody is not protective.
Thank you very much. And now actually I would like to invite Christoph, who asked a question, and I think it's better that Christoph, that you actually asked a question directly to Dr. Andreano, so I will unmute you. So Christoph, do you would like to ask your question? Looks like Christoph, can you hear us? Otherwise, I will read the question and maybe Christoph can comment afterwards. The question was that at the last Congress in October last year, we learned that T-cell immunity for only Wuhan vaccinated people was still 100% active for the circulating variants. What does the neutralizing antibodies really mean for protection against severe disease these days? With worldwide immunity over 99.5% against SARS-CoV-2, mainly hybrid immunity, or neutralizing antibodies really necessary?
Well, from all the literature, is actually, I believe, is quite necessary. It has been well shown that a delay on the production of the antibody response correlates with disease severity, and you need B-cell response also to control and to reduce the severity of infection. So while T-cell can guard initial response, can drive, of course, they are essential for the maturation of the B-cell response, and not only that, but I mean, it is known that this B-cell response, it is still necessary for protection.
Thank you. And I would also like to mention for everyone that if you have a question, you can also raise your hand and I will allow you to ask a question. Next up, we have Gregory, who asked a question, and I will unmute Gregory so you can post a question yourself.
Can you hear me?
Yes.
Yes, you can. Lovely talk, Emanuele.
Thank you, Greg.
Hey, I was just curious. I saw in your data there is an enrichment, progressive enrichment in the super hybrid immune individuals for class 4 antibodies among the monoclonals you cloned. Do you know what fraction, if any of them, are class 1-4? Do you see enrichment for class 1-4 antibodies in your panel?
Yeah, unfortunately, so it didn't occur to see a reduction in both signals for class 1 and class 4. We don't have any specific epitope covered by it. So because the antibodies that we use will put really much on the tip of the RBD, I'm not sure we will see, while for class 4 we use CR 3022. So we'll be really bottom left flank side of the RBD. I think the two are quite distant, and I'm not sure if with these two antibodies we are able to see class 1 to class 4 antibodies. So that could be a technicality. As for the enrichment of class 4, I mean, they are so rare. They are very, I mean, in our case, the way we single cell sort with the whole spike protein, but the fraction of these antibodies is so little. We really end up always with the 10 antibodies. I mean, the whole panel, probably there are less than 20 antibodies targeting the class 4. So they are very rare, but they are very broad. So they are good antibodies.
Exactly, of interest because they're so broad. Just not to belabor the point, but I think from ancestral Wuhan infection in the panel to your super hybrid, although they're still rare, it's like from almost, 1% only, up to like, I think 13% in your data set, like a 10-fold increase. Rare, but still, it looks to be a substantial increase.
Yeah. No.
Thank you for fielding my question.
No, my pleasure, Greg.
Thank you very much. And then we open up for Margaret Liu to pose her question.
Thank you very much, Emanuele. I just want to point out that Emanuele was one of the first winners of an ISV Travel Award when he was a trainee. So we are all very proud that of your development, and also how we were so prescient in picking you to get this award. But like to encourage other attendees at the meeting to see what can happen when you participate in the ISV meetings to help develop your career.
Great
At any rate,
thank you, Margaret.
this was a great talk and really nice work. But my question is, do you think that some of the issues for the specific germlines being activated were because of the antigen, the Wuhan antigen, something about it, or is it also mRNA as a vaccine modality? And the reason I ask this is, way back when we were doing early studies of DNA vaccines, comparing DNA and the inactivated influenza vaccines that were licensed vaccines. So this was studies in mice. But we found that whereas the inactivated flu vaccine resulted in stimulating responses that were then highly constrained by the by this original antigenic sin issue. So when you came back later on and you tried to immunize with a different strain, or in fact, they were constrained because of we didn't do all of this germline evaluation, but you could tell that the titers just weren't very good against a subsequent drifted strain. And yet with the DNA vaccine, you actually could then get very good responses even against a different strain. And so it makes one wonder whether or not in the case of a vaccine, that there could be two issues. There could be the actual antigen that somehow stimulates particular, germlines, or there's this sort of dominant epitope, or it could be that the modality is different. And I guess I would have thought that mRNA, we had always thought that it was because inactivated virus vaccines are very fixed in terms of their structure, because certainly for influenza, they're formalin inactivated. So it's just kind of, frozen in a structure. Whereas when you express the antigen by DNA, and I would have thought mRNA, you get a more flexible structure so that you could think that you might actually induce a broader array of antigen conformations, and therefore maybe of germlines. So it would be interesting to see what happens with, for example, in people who had gotten the adenovirus vaccines, to see whether or not they also had this limitation, and therefore, whether you think it's the antigen, the Wuhan, or maybe the modality. Thanks.
Yeah. Thank you very much, Margaret. Also for the introduction. Great meeting in Paris, a lot of fun. Please do apply to the scholarship and do attend the meetings. They are great. As for your questions, I really would like to split this in two. So the first thing is the differences between the vaccines, right? Now, inactivation, it is well known to lead to, if we take into account probably one of the most famous cases, the RSV case, right? Where inactivation of RSV basically led to the conformational switch of the fusion protein to pre-fusion and post-fusion, and then the vaccine wasn't effective. Actually, it was quite a disastrous clinical trials. But from that lessons, and thanks to the work that Jason McClellan and Barney Graham have done on the RSVF protein, they managed stabilizing the pre-fusion conformation, and that's basically what happened also in the case of the spike protein. So the ability to stabilize the S protein in its pre-fusion conformation really allowed to lead to the same antigenic sin that we have observed with infection with live virus. So because of these RNA vaccines and SARS-CoV-2 infection really pushed the same B-cell germlines. Now, the B-cell germlines 353 and 366 were seen that with a very low levels of somatic hypermutation. We're talking about even lower than 1%. They were already able, they were just there, ready to catch this virus, mature, and expand. They had these two motifs, one in CDR1 and one in CDR2, that even after 4 years, they are still conserved, still there, and they are pivotal for the binding of these antibodies. So again, because of the ability to stabilize the virus, to induce the spike protein, to induce the same antigenic scene, and because of the ability of the germline-like antibodies to be ready, we had this strong convergence, and independently from either you got infected or vaccinated, you've seen the same response. I'm 100% with you. I would love to see the same thing on different vaccines types. Adenovirus vaccines, for instance, will be very interesting, as well as inactivated vaccines will be very interesting. Unfortunately, we don't have these samples, but if you guys have these samples, we can definitely perform the same sort of analysis. I hope I have addressed all the points, Margaret.
Yeah, no, thank you. That's very helpful. Thank you.
Thank you.
Thank you very much. And then I would like to invite Engin Berber to pose a question.
Hi. Thank you for the presentation. Actually, I enjoyed it. So there are a few research studies that shows that as people receive the mRNA boosters, there is an increase in the proportion of the IgG4 level, especially in seronegative people, not in the pre-immune or seropositive people. So even though there is like no or less you see function retained in the IgG4, but still people are received a booster immunization with mRNA vaccines. There's a reduction in the hospitalization and the ICU admission due to severe COVID-19 infection. So I'm wondering, like, are those results like contradictory, or like is that antibodies are directed to the Wuhan strain, the original one?
Sorry, you mean in people that got multiple RNA vaccine doses, always the Wuhan vaccine, or is going to be an heterogeneous immunization? Because you will have a more of course a broader immune response if, for instance, you got Wuhan, then you got the dual Wuhan BA.5, then XBB.1.5. I mean, if you get subsequently vaccine doses with a heterogeneous spike protein, you will have a robust and broader immune response. And these eventually will lead to a reduction of severity of infection in ICU admission. While there is a certain ceiling that you will hit. I don't know if you came across the paper on Lancet about the guy that got over 250 doses of RNA vaccines. But I mean, you can with the same vaccine, you can boost into. I mean, there's gonna be a ceiling. That's basic immunology. You're gonna reach a ceiling. They've seen also an expansion of IgG4, but still really didn't change that much. But if you get a heterogeneous booster, that will definitely benefit your immune response. I hope I addressed your question.
Yes, actually, those are like heterogeneous immunization.
Then for sure, you're gonna boost that, and you're gonna broaden up your B-cell response. And interestingly, there are also other studies where it is true you always have this antigenic scene, but eventually, if you overexpose to Omicron variants, you will start seeing also other B-cell clusters expanding, of course. But the antigenic scene is going to be there, and the interesting thing is that the immune system will keep them up and restore them again and restore them again. And we found this story extremely interesting to follow across the last 3 to 4 years.
Thank you. There was another paper actually published, I think 4 or 5 years ago in HIV immunization. And they are claiming that the germline-centered vaccination is actually boosting neutralization and increasing the breadth of the antibodies. So, and they saw that the B-cell repertoire were increased. There was, I think, less than 0.5 and it's increased gradually up to 3% in the B cell population. So in your studies, have you also saw that similar pattern that the B cells expressing neutralization antibodies are increased, or is there no change?
So you're asking whether so can you just repeat the question, Engine? Sorry.
Yeah, the question is actually like, is there any increase in the B cell population that expresses the neutralization IgG, which broadly reactive, when people receive the heterogeneous vaccines?
Yeah. This is basically what we observed with HIV is completely different story there. You got these people that they got infected chronically for decades, and they start producing these broadly neutralizing antibodies, usually encoded by the VH1/2, very long CDR3. They break some of the checkpoints to try to put this very long CDR3 and start to be extremely cross-reactive. They have to overcome the glycan shield of the GP120, so they really need to push the immune system. And also the same donors will have hundreds of clades of HIV, meaning that the B cell response has to face so many different variants of the virus that really has to find a way. SARS-CoV-2 is complex, but not to that, luckily not to that level. But of course, when you start having this heterogeneous booster, you will have also a boost of B cell cross-reactivity against the different variants.
Thank
you very much. We have time for one last question, and I would like to invite Sita to pose your question.
Hello.
Hello.
A beautiful talk. Thank you so much.
Thank you.
I was wondering about the antigenic sin here, compared to what we see in the flu. And although like flu has been here forever and SARS-CoV is a new thing, and but we still see it. And so I was wondering if you would make any comments about what has been observed so far with the flu and limited time we had for the SARS-CoV-2 virus.
Yeah, sure. So it is true. I mean, we've been exposed to influenza since forever, and but there are some key differences. So first of all, you also find these sort of shared and publicly shared B cell clones. For instance, hemagglutinin influenza, very well known and studied are the IgHV 169. For influenza, they seems to be like a lot of them publicly shared, globally shared, and so you can see this convergence also in the case of influenza. Also in the case of RSV, there is the 169, the 118 that have been identified globally in different people across, in different countries, that we share some germlines that are possibly predisposed to certain glycoproteins. The huge difference is that for influenza, people do not have, or in this range, the same antigenic sin. They might be exposed to let's say, different strains of influenza for the first time when they are naive to it, leading to different antigenic sin, right? While for SARS-CoV-2, it wasn't the case, because we all were naive and we were all exposed to the same virus, leading to the same original antigenic sin. So you can find convergences, but with SARS-CoV-2, you definitely have this global convergence, which is extremely interesting to study for a B cell immunologist. Is extremely interesting to study.
Thank you.
Thank you, Sita.
So with that, I would like to finish this webinar by thanking you, Dr. Andreano, for a very nice speech today, and I hope to see you at the annual Congress in October. And I would like to thank all of you who listened and also posed questions very much today. And I hope to see you also again at the next webinar in May. So welcome back and have a good day. See you.
Thank you very much.
Bye bye.
Bye bye.